Good day to everyone. We are Team Samarium, and as a continuation of the video for the chromatographic techniques, we'll be presenting a comprehensive discussion on the various types of chromatographic methods of analysis. This video will cover the sample preparation, mobile and stationary phases, types of columns and detectors, as well as the quantitative applications of each type of chromatography. First is gas chromatography, which is used for analyzing volatile components in a sample with the boiling points and polarities of the compounds as the basis of separation. In GC, the mobile phase used is a chemically inert gas, which is called the carrier gas. Helium, argon, and nitrogen are commonly used in this instrumentation and is selected based on the instrument's detector. Other factors to consider is that for packed columns, the velocity of the mobile phase is usually 25 to 150 milliliters per minute, while the flow rate for the capillary column is 1 to 25 milliliters per minute. Since the separations in gas chromatography are based on boiling points and polarities, it is important to select a stationary phase with a high selectivity for the solute. Solutes with significantly different boiling points are easier to separate, otherwise, the stationary phase should interact selectively with the solute for efficient separations. In terms of the polarities, nonpolar solutes will be easier to separate using a nonpolar stationary phase and a polar stationary phase for a polar solute as per the like dissolves like rule. Aside from this, the stationary phase to be used must also be chemically inert so that it won't react with or degrade the solute during the separation process. It must be thermally stable as the operation of the GC is at high temperatures. It must also have low volatility and the appropriate polarity in order for the solute to separate. Shown in the table are the commonly used stationary phases for GC, with polymethylsiloxane as a good first choice in new separations, as its order of elution usually follows the boiling points of the solutes. If you'll notice, each type of stationary phase has a temperature limit. This is because at the stated temperature, the loss of the stationary phase is minimized and bleeding, which shortens the column's lifetime, is avoided. A schematic diagram of the gas chromatography is shown where the carrier gas is connected to a flow regulator, followed by the sample injection compartment, then through a column found in an oven before reaching the detector. Before introducing the sample to the instrument, a necessary preparation is first conducted for an efficient separation. Firstly, the sample must be volatile as non-volatile solutes may condense on the column, degrading the column's performance. In order to remove unwanted matrix components, several approaches may be used for isolating the analytes. One is the solid phase microextraction, where a solid adsorbent is coated on a fused silica fiber held within a syringe needle. The fiber is immersed into the sample by depressing a plunger exposed for a set time, then withdrawn into the needle and transferred to the gas chromatograph for analysis. It may be used in headspace analysis or direct immersion and is advantageous due to its efficiency, sensitivity, and it does not require a solvent for extraction. Another method is through the use of a purge and trap, where an inert gas is bubbled through the sample so that it reaches the adsorbent material or the trap. Then, the trap is heated while the carrier gas is continuously flushed to transfer the sample to the instrument. The next approach to sample preparation is what we call headspace sampling, where the sample is contained in a closed vial with an overlying space. A syringe will be used to take the volatile sample without touching the liquid and is then injected into the gas chromatograph. Lastly, thermal desorption can also be an option, especially for trace analysis of volatile organic compounds. The solid sample is placed in a glass-lined tube and secured with glass wool plugs, which then undergoes purging with carrier gas to eliminate oxygen and is heated. Upon heating, volatile analytes are swept by the carrier gas to the GC. This method uses cryogenic focusing, where solutes are concentrated at the column top by cooling the inlet below room temperature to enhance the separation efficiency. Aside from being volatile, the sample must also be present at an appropriate concentration. If the analyte is too low, it needs to be concentrated, such as in the process of thermal desorption and purge and trap method. However, the concentration must not be too high, as highly concentrated analytes may overload the column. Finally, to maintain optimal resolution and prevent pre-column band broadening, it's crucial to introduce a sufficiently sized sample in a small volume of mobile phase. A typical packed column injection port uses a rubber septum and a microliter syringe with the injector block heated at least 50 degrees Celsius above the highest boiling point in the sample to ensure rapid vaporization. On the other hand, capillary columns need special injectors. In the split injection, only 0.1 to 1% of the sample enters the column and the remainder is discarded, while splitless injections allow a higher percentage of the sample to enter the column, useful for trace analysis. For easily decomposable samples, on-column injection without heating may be used, with the column temperature slowly increasing for volatilization. Quantitative applications of gas chromatographs include environmental analysis, clinical analysis, analysis of the components in consumer goods, and in petroleum products. These are just a few examples as gas chromatography continues to find applications in diverse fields for quantitative analysis due to its high sensitivity, precision, and ability to separate and quantify a wide range of compounds. As a practice, you may pause the video to answer the problem applying GC in the analysis of determining the concentration of methyl salicylate in rubbing alcohol. A quick solution to the problem. First, the concentration of the methyl salicylate in the standard solutions must be determined. Then, construct a calibration curve with the concentration of the analyte in the x-axis and the signal of the chromatograph in the y-axis. From the calibration curve, the linear equation showing the slope and intercept will be used to determine the concentration of the methyl salicylate, which will obtain as 1.527%. However, this is not the final answer, as we need to account for the dilution by multiplying it to the dilution factor. This will give us a concentration of 1.9% methyl salicylate. The second type of chromatographic technique is the high-performance or high-pressure liquid chromatography. This technique is used for separating compounds that are soluble in a particular solvent where the elution order is based on the polarity of the analyte. The mobile phase used in HPLC is a liquid solvent or a combination of solvents that carries the sample through the chromatographic column. The polarity index may be used for selecting the mobile phase where a larger value denotes a more polar solvent. As for the stationary phase, a liquid film coated on a packing material with 3 to 10 micrometer porous silica particles are used. However, the stationary phase may bleed from the column due to its partial solubility in the mobile phase. For this reason, bonded stationary phases are covalently bonded to the silica particles. In HPLC, solutes elution order is determined by polarity. In normal phase separations, the least polar solute elutes first and the retention time is influenced by the mobile phase polarity. For this technique, adjusting to a less polar mobile phase extends retention times and improving the separation. Conversely, for faster elution, a more polar mobile phase is used. This is the case in reverse phase separation with the most polar solute eluting first. The illustration shows the schematic diagram for HPLC consisting of a mobile phase reservoir connected to a proportioning valve that allows the mixing of up to 4 solvents, a high-pressure pump that propels the mobile phase through the chromatographic column at a controlled rate, which is then connected to a pulse damper used to reduce pressure fluctuations generated by the reciprocating action of the pump. Then, in a loop injector, the sample is introduced before proceeding to the guard column, which acts as a filter to remove particulates that may enter the chromatographic column where the separation occurs. Finally, the separated components reach the detector. To highlight the sample introduction, the loop injector is interchangeable and available with volumes ranging from 0.5 microliters to 2 milliliters. It may be in a load position where the sampling loop is isolated and open to the atmosphere, and the syringe with a capacity exceeding the loop size is used to load the sample. Upon switching to the inject position, the mobile phase flows through the sampling loop, sweeping the sample onto the column. In some separations, a single mobile phase or the isocratic elution may not be suitable for all the solutes found in the sample. For this, changing the composition of the mobile phase during elution can be made in a single run. The quantitative applications of HPLC include the preparation of samples like wastewater and medicine for analysis, the quantification of food additives, preservatives, and contaminants in food and beverages, and especially in the field of forensic science. To practice analyzing problems involving HPLC, you may pause the video and answer the problem. In answering the problem, we can assume that both the spiked and unspiked sample will have the same amount of vitamin A as they only differ in a few decimal points. So the difference between the spiked and unspiked is mostly due to the standard vitamin A added. We can use the difference of the peak areas to solve for the unknown by equating the ratio of the amount of spike to its peak area to the ratio of the unknown and to its peak area. Solving for x would lead us to obtain 0.0210 milligrams. However, that is only for the 10.067 grams of serial sample. To express it as 100 grams of sample, we would also utilize the ratio of the unspiked to original amount and equate it to the unknown mass per 100 grams of sample. From this, the final answer would be 0.209 milligrams or approximately 0.21 milligrams of vitamin A in 100 grams of cereal.
Now, let us proceed to another type of chromatographic separation. Supercritical fluid chromatography is a chromatographic technique that utilizes supercritical fluids as the mobile phase. A supercritical fluid is a substance that is maintained at a temperature and pressure above its critical point where it exhibits properties of both a gas and a liquid. Carbon dioxide is the most commonly used supercritical fluid in SFC due to its favorable properties such as low cost, low toxicity, and environmental friendliness. SFC offers several advantages over traditional GC and LC techniques, making it a useful alternative for certain types of samples. For its mobile phase, a supercritical fluid is a substance that exists at a temperature and pressure above its critical point. At this critical point, the distinction between the liquid and gas phases disappears, and the substance exhibits properties of both phases. Specifically, it possesses densities and solvation properties that are intermediate between those of a gas and a liquid. For its diffusivity, in supercritical fluid chromatography, it refers to the ability of supercritical fluids to rapidly transfer molecules between the mobile and stationary phases. Supercritical fluids such as carbon dioxide above its critical point possesses high diffusivity due to their unique properties. This rapid mass transfer facilitates efficient interactions between the analyzed and the stationary phases, leading to faster separations and improved resolution. High diffusivity allows SFC to achieve high-speed analysis while maintaining excellent separation efficiency, making it a valuable tool in chromatography. For green chemistry, SFC is considered a green chromatographic technique because it predominantly uses carbon dioxide as a primary solvent. CO2 is a non-toxic, non-flammable, and environmentally benign substance. Additionally, carbon dioxide is readily available as a byproduct of various industrial processes, reducing the need for the production and disposal of hazardous organic solvents commonly used in other chromatographic techniques. By employing CO2 as the primary solvent, SFC minimizes the environmental impact associated with chromatographic analysis, aligning with the principles of green chemistry and sustainability. For the sensitivity, supercritical fluid chromatography, when coupled with sensitive detectors like mass spec, offers high sensitivity for trace analysis of compound. MS detection provides accurate identification and quantification of analytes with low detection limits, making it suitable for the analysis of complex samples containing trace levels of target compounds. The combination of SFC with MS detection enhances the analytical capabilities of the technique, allowing for the detection of analytes at concentrations as low as parts per billion or even parts per trillion. The high sensitivity makes SFC-MS ideal for a wide range of applications including pharmaceutical analysis, environmental monitoring, and food safety testing, where the detection of trace contaminants or active compounds is crucial for regulatory compliance and quality control. Other mobile phases and their critical temperatures and pressures are listed in Table 12.7. So let us proceed how SFC works. SFC operates through a series of steps from pumping of the mobile phase to the display of chromatographic results. Here's a simplified overview of the process. First is the mobile phase pumping. The process begins with the pumping of the mobile phase into the chromatographic system. In SFC, the mobile phase typically consists of a supercritical fluid, often carbon dioxide, along with modifiers or additives to enhance solubility and selectivity. The pump delivers the mobile phase at a controlled flow rate into the system. Next step is the sample injection. Once the mobile phase is flowing, the sample to be analyzed is injected into the system. This can be done manually or automatically using an autosampler. The sample is introduced onto the chromatographic column where separation of its components will occur. Third is the chromatographic column. The injected sample travels through a chromatographic column which is packed with the stationary phase. The stationary paste can be made of various materials depending on the separation requirements, such as silica, bonded pastes, or chiral selectors. As the sample moves through the column, its components interact differently with the stationary paste based on their physicochemical properties leading to the separation. Next is for the detection. As the separated components elute from the column, they pass through a detector. The detector monitors the eluent and generates signals proportional to the concentration of the analytes present. Common detectors used in SFC include ultraviolet visible spectrophotometers, refractive index detectors, and mass spectrometers. The choice of detector depends on factors such as the analyte's properties and the required sensitivity and selectivity. Next is the data processing and analysis. The signals from the detector are processed by a data acquisition system. The system converts the signal into digital data, which can then be analyzed and interpreted using chromatography software. The software calculates retention times, peak areas, and peak height, which are used to identify and quantify analytes present in the sample. Lastly, the display of results. The chromatographic results are displayed on a computer screen or chromatography workstation. The chromatogram, a graphical representation of the separation, shows peaks corresponding to individual analytes detected in the sample. The result may include information such as retention times, peak areas, concentrations, and any other relevant data. Throughout the process, the parameters such as flow rate, temperature, pressure, and gradient conditions can be adjusted to optimize the separation and achieve the desired analytical goals. The display of chromatographic results provide valuable information for researchers and analysts, aiding in the interpretation of sample composition and the identification of target compounds. Also, supercritical fluid chromatography has found many applications in the analysis of polymers, fossil fuels, waxes, drugs, and food products. Let us now proceed to the quantitative applications. A research laboratory is tasked with analyzing the concentration of pesticide compound C in a sample of agricultural soil using supercritical fluid chromatography. The laboratory employs a silica-based stationary paste and a mobile paste comprising carbon dioxide with a co-solvent ethanol, 95 by 5 volume by volume. To establish a calibration curve, the laboratory prepares a series of standard solutions containing known concentrations of compound Z ranging from 10 microgram per milliliter to 100 microgram per milliliter. Each standard solution is injected into the SFC system and the peak areas corresponding to compound Z are recorded. The following calibration curve is constructed. A sample of the agricultural soil is extracted and subjected to the same SFC conditions. The peak area corresponding to compound C in the sample is measured and around to be 7,000 arbitrary units. Calculate the concentration of compound Z in the agricultural soil sample. Now let's answer this question. To determine the concentration of compound Z in the agricultural soil sample, we can use the calibration curve constructed from the standard solutions. Since the calibration curve is linear, we can use the equation of the line to interpolate the concentration of compound Z corresponding to the measured peak area of the sample. First, we need to calculate the slope and y-intercept of the calibration curve using 2 points, which gives us point 1 that has a concentration of 50 microgram per milliliter and peak area of 6,200, while point 2 gives us a concentration of 75 microgram milliliter and peak area of 9,000. Now, to solve for the slope is equals to peak area 2 minus peak area 1 over concentration 2 minus concentration 1, which gives us 112 microgram per ml per unit. Using the slope and one of the points, we can calculate now the y-intercept. So, to calculate the y-intercept, peak area 1 minus m multiplied by concentration 1, which gives us 600. The equation of the calibration curve is peak area is equals to 112 multiplied by concentration plus 600. Now, we can use the measured peak area of the sample and solve for the concentration of compound Z, which gives us the concentration sample of 60 microgram per milliliter. Therefore, the concentration of compound Z in the agricultural soil sample is approximately 60 microgram per milliliter. Another type of chromatographic separation technique is liquid-solid adsorption chromatography, or also known as adsorption chromatography, is a technique used to separate components within a chemical mixture based on their interactions with an absorbent material. In this process, the mixture, which can be in the form of a gas or liquid, is passed over a stationary phase known as the adsorbent bed. The adsorbent material can be a solid such as silica gel or alumina with a large surface area capable of absorbing molecules from the mixture. The separation occurs as the components of the mixture interact differently with the adsorbent material. This interaction is based on various factors including polarity, size, shape, and affinity of the molecules for adsorbent surface. Components that have stronger interactions with the adsorbent material will be retained for a longer time, resulting in slower movement through the column, while components with weaker interactions will move more quickly. As the mixture travels to the column packed with the adsorbent material, the different components are selectively adsorbed and desorbed from the surface of the stationary phase. This differential adsorption and desorption lead to the separation of the components based on their individual characteristics. The elution profile obtained from adsorption chromatography typically shows peaks corresponding to each separated component as they are eluted from the column at different times. The retention time of each peak provides information about the interaction between the analyte and the stationary phase. By analyzing these peaks, researchers can identify and quantify the individual components present in the mixture. Adsorption chromatography is widely used in various fields such as analytical, chemistry, biochemistry, pharmaceuticals, and environmental analysis. It offers advantages such as simplicity, versatility, and the ability to separate a wide range of compounds with different chemical properties. However, it also has limitations, including the need for careful selection of adsorbent material and solvent systems to achieve optimal separation and the potential for irreversible adsorption of certain compounds. Overall, adsorption chromatography is a valuable tool for the separation and analysis of chemical mixtures in research, industry, and quality control applications. Next is the ion exchange chromatography. Ion exchange chromatography is a chromatographic technique used for the separation and purification of charged molecules based on their interactions with an ion exchange resin. In IEC, the stationary phase consists of a cross-linked polymer resin typically made of divinyl benzene cross-linked polystyrene. This resin contains covalently attached ionic functional groups. For its stationary phase, the stationary phase in IEC is a solid support made of a cross-linked polymer resin. The most commonly used resin is divinyl benzene cross-linked polystyrene. This resin provides a stable matrix with a high surface area for interactions with analytes. Covalently attached to this resin are ionic functional groups, which can be either positively charged or negatively charged. These functional groups are responsible for the ion exchange interactions with charged analytes. In IEC, the mobile phase plays a crucial role in facilitating the separation of charged analytes on the stationary phase. The mobile phase typically consists of a buffer solution containing ions that compete with the analyte ions for binding sites on the stationary phase. The mobile phase in ion exchange chromatography serves to facilitate the competitive exchange of ions between the analytes and the stationary phase, allowing the separation of charged molecules based on their interactions with the stationary phase functional groups. By carefully selecting the composition and conditions of the mobile phase, analysts can optimize the separation process and achieve the desired chromatographic results. Lastly, in size exclusion chromatography or SEC, the stationary phase consists of micron-sized particles with porous structures. Two common types of stationary phases encountered in SEC are silica particles and cross-linked polymer resin beads. Both materials possesses pores that allow for the separation of molecules based on their size. For the silica particles are commonly used as the stationary phase in SEC. These particles have a porous structure with a ranging size from approximately 50 to 4,000 angstroms. The porous nature of silica particles allow molecules to enter into the pores based on their size. Larger molecules are excluded from entering the pores and are eluted from the column first, while the smaller molecules can enter the pores and take longer to elute. For the cross-linked polymer resin beads such as divinyl benzene cross-linked polystyrene resins are another type of stationary phase used in SEC. These resin beads also have a porous structure with a pore size ranging from 50 to 1 million angstroms. Similar to silica particles, the pores in cross-linked polymer resin beads allow for the separation of molecules based on their size. Larger molecules are excluded from entering the pores and are eluted first, while smaller molecules can penetrate the pores and have longer retention times. In SEC, which is also known as molecular occlusion chromatography or gel permeation chromatography, the separation mechanism is based on the solute's ability to enter into the pores of the column packing. Larger molecules are unable to enter the pores and are excluded from the stationary phase, resulting in shorter retention time and earlier elution from the column. In contrast, smaller molecules can penetrate the pores and spend more time within the porous structure, leading to longer retention times and later elution. Overall, SEC is a valuable chromatographic technique for separating molecules based their size. By utilizing porous stationary phases such as silica particles and cross-linked polymer resin beads, SEC can effectively separate molecules across a wide range of sizes, making it useful in various fields such as biochemistry, polymer chemistry, and pharmaceutical analysis. The size of selectivity of a particle packing is not infinite, but is limited to a moderate range. All solutes significantly smaller than the pores move through the column's entire volume and elute simultaneously with a retention volume of VR is equal to Vi plus Vo, where Vi is the volume of mobile phase occupying the packing material's pore space, and Vo is the volume of mobile phase in the remainder of the column. The maximum size for which equation holds is the packing material's inclusion limit or permeation limit. All solutes too large to enter the pores elute simultaneously with a retention volume of VR is equals to Vo, which the equation defines the packing material's exclusion limit. In between the inclusion limit and the exclusion limit, each solute depends on amount of time in the pore space proportional to its size. The retention volume for a solute is VR is equal to Vo plus dVi, where d is the solute's distribution ratio, which ranges from 0 at the exclusion limit and 1 at the inclusion limit.
Chromatographic scientific second its comprehensive history. Chromatographic columns are part of the exploration series of the field. So five chromatographic methods that are uses columns are these chromatography, liquid chromatography, ion exchange chromatography, size exclusion chromatography, and chiral chromatography. So the basic principles of the chromatography can be applied to the five methods. So first is the gas chromatography. So the northern phase is the gas. So each columns are usually and slow, and the thickness of the stationary phase ranges between 0.5 micron, and the thicker the layer, the more volatile the okay. So the table shown summarizes the use of different columns. Pack columns can only achieve about 50% of the efficiency of a compound, all coated by tubular, and furthermore, the diatomaceous earth packing is rapidly due to the semi-permanent absorption of impurities in the column. In supercoated open tubular columns, the columns are first coated with a thin layer of sorbent solid, such as diatomaceous earth. In contrast, fused silica wall and SWC open tubular columns are manufactured to be virtually free of these adsorption problems. So here are generated images of fused silica columns that is specialized for measuring synthesis, and another is for specialization to withstand extreme heat. So next is the right liquid chromatographic columns. This liquid chromatography uses the liquid mobile phase. So three basic types of liquid chromatography columns are liquid solid, and ion exchange. So in terms of their columns, usually HPLC and Zinc ahead of the analytic column and extend the lifespan of the column. So the guard column is particulate matter, contaminants, and molecules that bind irreversibly to the column. The guard column is a stationary similar to the column. As for its material, HPLC columns are usually packed with molecular porous particles. So these particles made from polymer or glass beads, and these particles are surrounded by a thin layer of silica, resin, or other type of ion exchange resin. As for resin particles, it is usually made up of silica. So the high-performance liquid chromatographs are commonly from stainless steel, which has a length of 3 to 25 centimeter and a diameter of up to 5 meter, which is efficient, which the efficiency of the columns can be increased. So moving forward is the ion exchange chromatographic columns. In ion exchange chromatographic columns, this use to separate ions and molecules that can be easily ionized. Ion exchange chromatography is often a form of column chromatography. So as ion exchange is based on the electrostatic interaction of charged surfaces, the general principle of separation of ion exchange chromatography is by reversible exchange of ions between the direct ions present in the sample solution to the ions present on an exchanger. So this process, types of exchangers, the cation exchangers can be used, in which they contain charge groups on the surface and of the matrix. So the charge groups of the matrix is surrounded by ions of the opposite charge and suspended in an aqueous solution. In this ion cloud, ion can be reversibly exchanged without changing the nature and the properties of the matrix. So depending on its application, the following stainless steel, polyether, ketone, titanium, glass, that can vary in diameter from 2 mm to 5 cm, and a length 3 cm and 50 cm purposes. And the common packing materials for ion exchange columns are alloys, sulfur, seal, diatomaceous earth, and alloys. Further, it's the size exclusion of columns. So this separates molecules based upon their size and not based upon their molecular weight. Shown in the figure is the schematic of the size exclusion column. The larger particles will get first because they are too big to fit inside the pores, wherein the smaller particles will get last because they fit very well inside the pores. So molecular sieves are a typical packing material for these columns. One often used molecular sieve is zeolites. So large molecules cannot pass through the pores of the molecular sieves, while small molecules can. Because of this, bigger molecules are able to move along the column more quickly than smaller ones. Other packing materials for silica and polysaccharides and other polymers are used in size exclusion chromatographic columns. For size exclusion separation, the pore size ranges from 4 to 2 and below. Last, Lastly are the chiral columns. So chiral columns are used to separate enantiomers, and the separation of chiral molecules is based upon stereochemistry. A quick recall on what enantiomers are. These are two compounds with the exact same connectivity that are mirror images of each other, but that are not identical to each other. So going back to chiral columns, these columns have a stationary phase that selectively interacts with one enantiomer over another. These types of columns are very useful for separating mixtures. Two approaches to the separation of chiral compounds is first, the chiral stationary phase. So the chiral stationary phase prepared is prepared by binding the chiral selector supported, usually silica. The chiral stationary phase is commonly classified into type to type 5 according to their chiral recognition mechanism. Type 1 is the solute chiral stationary phase complexes formed by attraction. Type 2 is the solute CSP complexes formed by attractive interaction with emission complexes, also play important roles. Type 3 are the complexes of solute CSP formed as inclusion complexes in chiral cavities. Type 4 is the solute CSP complexes formed as metal diester complexes. And type 5 is the CSP is a protein and the solid CSP complexes formed in these cases are a result of polar and hydrophobic interaction. So another one is the chiral base. So chiral mobile base is used as an alternative for relatively more expensive chiral station phases. And in this technique, the additives dissolved in the mobile base form transient complexes with the solid.
Now that we know the different types of chromatography techniques, how we prepare the samples depending on which type, and the different columns that comes with it, it's now time to venture into the different detectors that this instrument is using. Let's start with gas chromatography. The first on the list would be thermal conductivity detector, or also called as TCD. Now TCD is one of the earliest detectors and is considered a universal detector for GC. TCD measures any GC sample component having a thermal conductivity that is different with that of our carrier gas. It uses a single filament in a constant power DC supply and requires only one gas type to operate, either helium or hydrogen, since they are the one that have the highest thermal conductivity, but sometimes others uses nitrogen or argon, as can be seen in this table. Usually TCD is used to detect inorganic gas and other components. The way TCD works is like this. When a voltage is applied to a filament, what happens is that it heats up. The steady-state temperature depends on the applied voltage, the resistance of the filament, and the rate to which the filament loses heat to the surrounding. If a filament is immersed in a gas stream, any changes in thermal conductivity of the gas causes a change in filament temperature, thus changing the resistance. A rapid switching valve causes it to sample the carrier effluent and the reference gas alternately. If the two gas streams are identical and no resistance change, this means that we have no component present. However, when a component enters the detector, the filament temperature drops when the column flow is switched in and then recovers when the reference gas is switched in. The electronics senses this change and adjusts the power to the filament to keep the temperature constant. The power demand curve amplitude depends on the thermal conductivity difference between the column flow, where a component is present, and the reference gas. This is then plotted and ends up in a chromatogram. The next detector will be flame ionization detector, or also called as FID. The FID is the most commonly used general purpose GC detector. As the sample exits the column, it is burned in a hydrogen air flame and ionized particles are measured as an electrical current. The way FID detector works is that you create a hydrogen flame by burning air and hydrogen that is supplied from below. The carbon in a sample carried into the detector on a carrier gas is oxidized by this hydrogen flame, which causes an ionization reaction. The ions formed are attracted by a collector above the flame to an electrostatic field, wherein the components are detected. Additionally, the number of ions that is produced is proportional to the number of reduced carbon atoms in the flame. Hence, this technique is a mass-sensitive destructive technique. The FID is the most common detector used in gas chromatography as mentioned earlier. The FID is sensitive to and capable of detecting compounds that contains carbon atoms, which accounts for almost all organic compounds. However, FID is not sensitive to carbon atoms with a double bond to oxygen, such as in carbonyl groups and carboxyl groups, to name a few. The third detector will be electron capture detector, also called as ECD. This detector is one of the most widely used detectors for environmental samples because this detector selectively responds to halogen-containing organic compounds such as pesticides and polychromated biphenyls. Now, in this detector, the sample eluates from a column is passed over a radioactive beta emitter, usually nickel-63 is used, and an electron from the emitter causes the ionization of the carrier gas, which is sometimes nitrogen, and the production of a burst of electrons. In the absence of an organic species, a constant standing production of current between a pair of electrodes results from this ionization process. The current decreases markedly, however, in the presence of organic molecules containing electronegative functional groups that tends to capture the electrons, hence the name electron capture detector. Compounds such as halogens, peroxides, quinones, and nitro groups are detected with high sensitivity. The detector is insensitive to functional groups such as amines, alcohols, and hydrocarbons. ECD are highly sensitive and have the advantage of not altering the sample significantly, in contrast to FID. So the picture in this slide, particularly this one, is an example of a ECD detector that uses nickel-63 as the beta emitter. So we have next is flame photometric detector or FPD. FPD detects organic compounds containing sulfur or phosphorus. So you have your sample, they are burned in a hydrogen flame producing light. So this is the same with that of FID. An optical filter is used to make the detector specific to sulfur compounds or phosphorus. The filtered light is then detected using a photomultiplier tube, and after that, it will be sent to a data processor which produces the chromatogram. So the picture here shows an example of the instrument. Sulfur generally responds to the square of the sulfur atom concentration, meanwhile for phosphorus, the response is more linear. For our last detector, we have the flame thermoionic detector or FTD. Now, FTD is a selective high-sensitivity detector for organic nitrogen compounds and inorganic and organic phosphorus compounds. The selectivity of the FTD for phosphorus compounds is not as good as that of the FPD, and the FTD does not react to inorganic nitrogen compounds. This detector works by, we have your sample, and the sample is passed through a glass bead. In this case, it is rubidium sulfate. This rubidium sulfate is electrically heated in the presence of hydrogen and air, and this forms a plasma around the bead. Now, if your sample came in contact with this bead, what happens is they transform to an ion, and a collector will collect those ions and send it to a data processing machine, which forms the chromatogram.
For the detectors for HPLC, majority of detectors are not unique to high-performance liquid chromatography. The detectors for HPLC are mostly standalone instruments of measurement or modified versions of the standalone instrument. The spectroscopic detectors work by measuring the absorbances of solutes as it elutes out of the column, where we consider a setup like this where the solutes absorb at certain wavelengths. The first detectors to discuss under spectroscopic detectors is the UV-vis spectrophotometer. UV-vis detectors work by passing visible and UV light through a sample which elutes from the column in a flow cell. The chromatogram shows absorbance over eluted time. Higher absorbances correspond to higher concentrations of solutes that absorb at the chosen wavelength. The limitations, however, of this detection method is that the mobile phase does not absorb strongly to avoid interfering with the analyte signals. A diode-array spectrophotometer records the entire spectra and produces a three-dimensional chromatogram. This allows the detection of absorbances along different wavelengths, beneficial for when analytes and solutes absorb at different wavelengths. Next are fluorescence spectrometers. Fluorescence spectrometers detect analytes that fluoresces. This provides additional selectivity since fewer solutes are capable of fluorescing. The resulting chromatogram, therefore, is a function of fluorescence intensity. The next set of HPLC detectors are based on electrochemical measurements. In general, electrochemical detection involves the use of an electrode to detect and quantify analytes in the sample. As the analytes elute from the HPLC column, they flow into the electrochemical cell. If the analyte then is oxidizable or can be reduced, it undergoes oxidation or reduction at the electrode, leading to the transfer of electrons. This transfer of electrons generates a measurable electrical signal, typically a current or voltage, which is directly proportional to the concentration of the analyte. For the first type of electrochemical detection, amperometry is based on measuring the current resulting from the oxidation or reduction of analytes at a constant applied potential. It is highly sensitive and selective for electroactive compounds and are thus applicable to a broad range of analytes, but it is limited to these compounds. It also runs the possibility of being interfered by non-electroactive compounds. Another limitation to all electrochemical detection methods is the wear and tear of electrodes and the frequent need for replacement, especially for complex sample matrices requiring electrolysis of different compounds. Now, while amperometry measures the generated current, coulometry measures the electricity required to convert the analyte. This is advantageous for applications where measurement of charge is necessary, but it lacks specificity when conversion products of multiple analytes overlap. Also, it needs strict calibration requirements. While coulometry and voltammetry deals with measurements from constant potentials, voltammetry includes measurements from a potential scan. Similar to diode array spectrophotometers, this generates a three-dimensional chromatogram that shows generated currents across a range of potentials. This is advantageous due to its versatility, but it is limited by the complex specialized instrumentation required and its sensitivity to impurities and other electroactive species. Refractive index detectors measure differences of refractive index between eluted analyte and a reference stream that only contains the mobile phase. This is similar to comparisons between a sample and a blank in a spectrophotometer. This is universal and responds to all compounds. However, it is not useful for gradient elution where mobile phase composition is varied, unless the mobile phase gradients have the same refraction index. Lastly, in HPLC-MS, the two techniques are connected by an interface that transfers the separated components from the liquid chromatograph column into the mass spectrometer ion source. This provides additional structural data ideal for identifying unknown analytes. However, the interface for HPLC-MS is more difficult versus GC-MS because of the liquid mobile phase and its incompatibility to a high vacuum system.